Okay, this one is going to be on molecular testing in blood bank, and we're not going to go super in-depth to this because that's going to be like more empty questions if you're getting questions on it. I mean, like past that PCR is an amplification technique. I just don't think you're going to get a whole lot of questions on it. And if you work at a facility that needs to run PCR testing, then they will train you more in depth, more properly. Okay, so molecular testing in blood bank. And like I said, this is going to be just like barely scratching the surface, maybe more like blowing the surface. So I wanted to start it with a picture for some reasons that you would even be running molecular testing. Okay, so your applications for molecular testing in blood bank, transplant, HLA antigen level and allele level typing for HPC and organ transplants. I know we've talked before just a little bit about how your blood needs to have like have to have certain matches, and then organs have to have like your HLA more specific next level, you know, matching. It can't just be like the same blood type. So HLA antigen level and allele level typing for HPC and organ transplants. And in fragment studies for HPC transplants. And then your transfusions, red cell typing in multiply transfused patients. Determine the blood type when the DAT is positive. Complex RH genotypes and weak expressions. Screen for antigen negative donor units when anti-sera are unavailable. Donor antigen screening for prevention of alloimmunization. Means, is that from yourself or from someone else's antigens?
Say that again.
Alloimmunization, is that going to be from yourself or from someone else's antigens? Right here, alloimmunization.
From someone else?
Correct, from someone else. And then if it was from yourself, it would be auto. But this is allo, so that's from someone else. HDFN, hemolytic disease of the fetus and newborn, to determine parental RHD zygosity. That basically means to determine the dad's RH. So, you know, if mom's D negative, that there's a potential, and she has a potential D positive baby, that can cause problems. So to determine the parental RHD zygosity is pretty much determining what the parents are as far as there are. Just a moment. I don't know what it did. Is it still sharing? Can you see me or can you see the page?
We can see you.
See? Okay. I don't know what happened. Let me get back to it. Okay, sorry. Now, donor testing, you detect the virus in the donors that may be below detectable levels by antibody detection methods. So that's a really awesome application, is that they can see if there's even like little single multiples of the DNA for the virus. And then relationship testing to establish paternity and legal relationships for immigration. So ensuring that someone who has, you know, brought a child across the border is indeed the relationship that they say they are to them. And that kind of helps prevent people from stealing kids or purchasing kids or any such form to get across. Oh, and I'm sorry, your HPC is a stem cell transplant. Okay, so that's just the kind of a basis for like why you would be doing any kind of molecular testing. Now, the first one we're going to talk about is PCR. You all know what that stands for?
Oh, right.
It's going to be polymerase chain reaction. And this is an amplification technique.
And this is some kind of testing?
Yes, this is a, well, it's a lab test, but it is molecular diagnostic testing. But what it does is it rapidly multiplies specific DNA sequences. So if you have a single DNA strand, let's say your virus is too low to be detected with your normal lab test. So if they amplify it by this polymerase chain reaction, the PCR, then it multiplies it, and then you can test it and see if it's there in that specific case. Does that make sense?
Yes.
So rapidly multiplies specific DNA sequences. So the first step is the DNA is denatured. And now it can be denatured in a few different ways. The best way to remember it is denatured by the C. So your S stands for salt, referring to high ionic strength salt. And then the second one is E, by elevated temperature. And then your A is going to be an alkaline pH. And again, I wouldn't, necessarily remember all these. This was just a fun way to remember how it can be denatured. But generally, I don't think you'll get asked a question past, PCR being an amplification technique, or that it multiplies DNA sequences. So the DNA is denatured, and then a primer is added. And this is to attach specific areas of DNA. And then a primer is the definition for it, is a short segment of single-stranded DNA used to initiate DNA replication in PCR. So short segment of single-stranded DNA, and that is used to initiate DNA replication in PCR. Oh, and this is like a it is like a single strand template. Single strand. More basic terms. I thought that was an easier way to put it. Single strand template. And then next, your DNA is amplified and replicated. Oops, sorry, this should have been on a... I hate when it does this. Just a moment. Okay, so your DNA is amplified and replicated. So each one of these cycles doubles the amount of DNA. And I'm going to highlight our processes really quick. So it rapidly multiplies specific DNA sequences. First, it is denatured, and then the primer is added, and then it is amplified and replicated. Okay, now there's PCR based HLA typing procedures. And these, I'm just going to give you all the names and their acronym for them. Because again, I just don't think you'll get asked on any specifics on them. But it's good to know that they are all HLA typing procedures. So the first one is SSP, and that stands for sequence-specific primers. The next one is SSO, and that one's sequence-specific oligonucleotides. Next one is SBT. That one's going to be sequence-based typing. And then the last one is STRs. And that one's short tandem repeat.
We did get asked, give me a minute. What was that one? Here, we did get asked something about the, I think it was the SBT. Let me look here. Yeah, in the molecule, it says in molecular techniques for HLA typing, which high-resolution method analyzes nucleotide sequences to define the allies? And it says it's SBT.
Oh, okay. I'm surprised they asked one on it. And I think, well, then definitely if you ever put that on the test, it would be that exact question.
Okay.
And then these help in the early detection of transfusion-related pathogens.
Now, when you mean early detection,
it means that you can, through PCR, you can detect the pathogen or the virus that could cause harm to someone else before it's detectable with normal tests. So, like a big one is HIV that you can get through blood products, correct? So, your normal HIV test, let's say it can, oh, like that's a really good example because you know how people they get tested and then they ask you to come back in a few months or 6 months or every month, depending how many tests you've gotten done so far, and they have you retested. Or like the flu, if you test negative, they might call you back in a week and test you again. So it means that the titers aren't high enough yet to detect for that lab test to detect the virus. So through PCR, they amplify it, or they amplify it and multiply the DNA sequences so it's detectable at an earlier rate.
Okay.
Now the next one is bead chip technology. So the speed chip technology, you have oligonucleotide primers attached to the colored beads on a slide. So oligonucleotide.
What is that oligonucleotide mean?
Let's see. Oligonucleotide is a polynucleotide whose molecules contain a relatively small number of nucleotides. And let me see.
Okay.
And your nucleotides are a compound consisting of a nucleoside linked to a phosphate group. Nucleotides form the basic structural unit of nucleic acids such as DNA. So a few nucleotides are just a few of the basic structural units of DNA.
Okay.
That's a very good question. Primers attached to colored beads on a slide. And we found that that one is small. Now, forms a basic structural unit of a nucleic acid such as DNA. Okay, so the oligonucleotide primers attached to the colored beads on a slide, and then amplified and digested DNA. They bind to primers. And again, primers are short segments of single-stranded DNA used to initiate replication. And then after that, computer analysis determines which primers attach.